Roshni. Can you close the door, actually? Okay, good afternoon, everybody. Today, our next speaker is Dr. Cerys Manning. Dr. Manning is a stem cell biologist from the University of Manchester. She has completed her master's degree in biochemistry from the University of Oxford, where she received the Mary Somerville Prize for obtaining a first-class degree. She completed her PhD in tumor cell biology from the University College London. Here she used intravital imaging of mice to compare signal activation between non-motile and invasive cells during tumor growth and metastasis. Next, she moved to the University of Manchester as a postdoctoral research associate in 2013. Dr. Manning has received many grant awards. She got her own Sir Henry Wellcome postdoctoral research fellow grant in 2015 to lead and manage her own project. Here she worked closely with mathematicians to understand gene oscillations during neural development. She showed for the first time in the developing tissue environment that single cell expression dynamics of the notch target HES5 are noisy in stem cells and can change over time between random fluctuations and pulsatile oscillatory behavior. These oscillatory or random fluctuations in HES5 expression correlated with final neural subtype fate. In 2019, she won the University of Manchester Presidential Fellow Award and started her independent career collaborating with clinical geneticists. Currently, since 2021, she is a Medical Research Council Career Development Fellow and is leading her lab group to integrate dynamic gene expression with mechanical signals to understand retinogenesis and ocular disorders. Today, Dr. Manning will talk on modeling developmental eye disorders in human retinal organoids. Ladies and gentlemen, please welcome Dr. Cerys Manning. Dr. Manning, stage is yours.
Thank you very much for that kind introduction. So, as was mentioned, my background is kind of in neural development, but now I've set up my lab interested in how eyes form during embryonic development and how that can sometimes go awry. And so some babies are born with congenital eye disorders that severely impact vision. And we use a number of models in the lab, transgenic mice, zebrafish, and human iPS stem cells. And today I'm going to talk to you about our human stem cell work and our use of retinal organoids. Okay. For some reason, here we go. So the kind of key question, an overarching question that I'm interested in is how during eye development, retinal progenitor cells, so these are kind of stem cells that can either proliferate, generate more copies of themselves, enable the growth of the tissue, how they decide between decisions of proliferation versus differentiation to generate all the types of mature cells that we have in our retina. So for example, the light sensing photoreceptors, but other neurons and glia in the eye. what we really want to understand is how these retinal progenitor cells balance proliferation and differentiation. And these decisions are crucial during eye development, because if they somehow become imbalanced or go awry, then this can lead to developmental eye disorders. So we know that there are multiple signals that the stem cells are receiving. They might be soluble, such as growth factors, for example, soluble BMPs. They may be mechanical stimuli, biophysical cues coming from the extracellular matrix or surrounding cells. And these kind of external cues are integrated with the current intrinsic state of the gene network to then enable decisions between proliferation and differentiation. And during eye development, as these cells are deciding between proliferation and differentiation, there's actually a large amount of tissue shape changes that are occurring. So, for example, early in eye development, this kind of brown stuff is the developing brain. The eye tissue starts to evaginate and grow away from the developing brain towards the outside of the embryo, the surface ectoderm. As this newly formed optic vesicle starts to touch the outside of the embryo, there's then signaling between this tissue layer and the early optic cup to generate the lens, the early lens, and what will become the developing kind of retina. This dynamic signaling between the early lens and the early retina causes evagination. So this is then tissue movement and cell movement inwards back towards the brain, and creating a kind of cup-like shape, which essentially then there's increased cell proliferation and differentiation, and then you have the final layers of the developing eye that we know, the lens, the neural retina, which is the light sensing, which contains the light sensing cells, and this yellow layer, which is the retinal pigmented epithelium, which supports and maintains the photoreceptors and the light sensitive cells. So there's kind of morphological events, for example, these outward migration of cells, inward migration here, and this is happening at the same time as cells are undergoing fate decisions, for example, proliferation. There's patterning between, or there's specification between different layers of the retina, and also differentiation. These two processes happening at the same time, morphogenetic kind of tissue shape changes and cell movements, as well as cell fate decision between proliferation and differentiation. And if we think about the retina specifically, there are kind of some key features. We have retinal progenitor cells or retinal stem cells, which are formed early, and then they can differentiate into the 7 main cell types of the retina. And the first cells that are generated are these ganglion cells. And then there's kind of 2 waves of differentiation. The first wave of differentiation, you have these 4 cell types, including the cone photoreceptors, which are responsible for our color vision. And then a later wave of differentiation, which also includes the rod photoreceptors, which are the kind of highly light sensitive cells that enable like night vision or vision when there's low light conditions. And these main cell types are highly organized in the retina. And so we have these different layers. For example, the outermost layer is where the photoreceptors sit. Here you have the cones in green and the rod cells in purple, and then in the kind of have some central layers that contain kind of multiple different neuronal subtypes, and then the innermost layer are the ganglion cells, which are essentially receiving signals from these neurons and taking those signals to the brain. So you have a highly kind of organized differentiation time, but also kind of spatially organized cell types in the retina. And this will become important when we talk about retinal organoids later. But what we're trying to understand is like how do the retinal stem cells make decisions between proliferation and differentiation in these different cell types, and what processes may go awry in babies that are born with developmental eye disorders. And the eye disorders that we're particularly interested in are coloboma. So this is a morphological disorder where the eye hasn't actually finished kind of undergoing its shape changes. And actually there's a fissure, so a small gap in the eye, which can be seen by this kind of keyhole pupil shape. And the second disorder that we're interested in is called microphthalmia. Then patients essentially have a small eye, which you can see here. And this may be because the retinal progenitor cells have not undergone enough proliferation, potentially they've undergone apoptosis, or there's been premature differentiation. So the balance between proliferation and differentiation has maybe has gone awry. And so this is a kind of disorder of cell fate, and we're particularly interested in this one. But interestingly, these two disorders, so this morphological disorder and this kind of disorder of cell fate and cell fate decisions, often occur together. And we're wondering whether they occur together because actually there's a feedback between tissue morphogenesis and kind of tissue shape changes with cell fate decisions in the eye. And so that's kind of our hypothesis, and really our overarching question is how do the misregulation of cell fate decisions contribute to these developmental eye disorders? So one of the ways that we'd like to try and understand these disorders is to use human models. Okay, so in eye development, we're lucky to have a human retinal organoids, and we'd like to try and use these as a model for human retinal disorders. So one of the great things that we have is that these stem cells or the retinal organoids are actually human derived. So we can match the exact genetic changes seen in the patients. And so we can generate these starting with iPSCs or human embryonic stem cells. We then plate a specified number in each well of a 96 well plate, and then undergo kind of media changes over the course of a large number of days. You can see that by the end we're up to like week 22. And interestingly, the time scale of this organoid generation is actually quite similar to actual human eye development in human gestation. So I'm not kind of going to go into too much of this and what kind of media we use, but initially we kind of push the stem cells into a more neural state, and then make sure this is like anterior neural, and making sure that we kind of define them into retinal stem cells rather than forebrain stem cells by adding BMP4, which is a key factor in retinal development. And then once we kind of have added this, then we keep them in a retinal differentiation media, and they start to form these little kind of optic vesicles, which is a bit like what we saw in the schematic of eye development, where the cells start to kind of, the forebrain starts to exude in these evaginations to create these optic cup-like structures. And then you can see over a course of time that they kind of mature into a more kind of smoother nodules or kind of optic cups, and undergo differentiation to generate the retinal cell types that we see. So again, just to remind you, we have these 7 retinal differentiated cell types, and we have these layers. So can the retinal organoids recapitulate these features that we see in the human retina? So basically, in short, yes, they can. At day 30, we see that these early retinal organoids are mostly retinal progenitor or retinal stem cells, and these VSX2 and SOX2 are kind of markers of the stem cell state, and of the retinal stem cell state, I should say. And actually, at this stage, most of these organoids are these progenitors. If we look a bit later, day 40, we can identify proliferating cells using a phosphohistone H3 antibody. And we find these dividing cells at the surface of the organoid. And this is really interesting because also in human fetal tissue, but also in animal models, the dividing cells always divide at the apical surface. And so this seems to be recapitulated in our retinal organoids. And we're also starting to get differentiation. So this green marker here is a marker of the retinal ganglion cells, which, if you remember, a couple of minutes ago, I said these were generated first. And these are our first differentiated cell types that are generated. We can then look again a bit later, and we can find that markers for the photoreceptors can then be observed, as well as having this brain3 marker of our retinal ganglion cells. So we're able to generate a lot of all the cell types that we find in the human fetal tissue. And interestingly, if we stain the fetal tissue here on the left for markers of different cell types, so this in light blue here is for the photoreceptors, this pink is for actually for amacrine cells, but the neurons that I'm telling you that were basically feeding the signal from the light responses photoreceptors towards the ganglion cells, which are in yellow. You can see there that these are these marker staining different layers that I talked to you about, and this is just a slightly later. Just a different region of the retina. You see this photoreceptors on the outside, the ganglion cells in yellow on the inside, and then this pink marker for the neurons in the middle. And you can see in the human iPS-derived retinal organoids, you also see this same structure in the photoreceptors on the outer layer, the ganglion cells in the inner layer, and the kind of pink is somewhere in the middle. It's not quite as clean as and beautifully layered as the fetal retina, but it's still we haven't really done anything at all here. We just created a ball of cells and changed media. The cells themselves have generated the layers, of basically self-organized into these layers. So the iPSC-derived human retinal organoids can generate the cell types that we find in the fetal eye, as well as in the same kind of structure and organization. But how about the gene expression? These are just some markers, but are they really truly like the gene expression of the cells that we find in the fetus? So people have done single-cell RNA-seq from the human fetal retina taken from day 82. And so this single-cell RNA-seq, basically you're able to capture gene expression of individual cells, and then using clustering, you can kind of cluster similar cells based on their gene expression. And you can find clusters of cells that have similar gene expression, and then look at their genes, look at what genes are expressed in these, and kind of annotate them based on known markers. So we can find the same cell type clusters in the human fetal retina as we can see in single cell RNA-seq from the organoids. So although these UMAPs here are not the same shape, we can find the same clusters, for example, here, the retinal progenitors or the retinal stem cells in the fetus, as well in the retinal organoids. We can find the photoreceptors and the retinal ganglion cells. So this is just highlighting a few. But what people have also done is then to try and combine these data together to really do a better analysis of whether the gene expression is similar between the cell types generated in the organoid and in the developing fetus. So this is now basically clustering both single cells from the organoids and the human fetal retina. And interesting, they're from kind of the same time point. So day 90 from the organoids and day 82 from the fetal embryos. So you can see that they actually generate, again, these clusters that we expect, the progenitor cells, various different photoreceptors, retinal ganglion cells, and the different kind of cell types that the differentiated cell types that we see. But what's kind of interesting to look at is these two plots. So you can see these shapes. This shape here is the same as this is here. Okay, and the cells that have come from the organoids are shown in purple, and the cells that come from the fetus are shown in light blue. And what you'll see is there's really nice overlap. For example, the progenitor cluster here, which is mapped onto here, these contain both fetal cells and cells from the organoids. So we can say that the gene expression of cells in the retinal organoid really closely matches the gene expression of the cells from the fetus. And we can then kind of conclude that the retinal organoids are a nice model for human retinal development. And it also, sorry, I just missed it out, but if we look at the composition of the different 7 cell types that I was talking about, the retinal ganglion cells, for example, and the photoreceptors, what you can see that is relatively similar between the fetal retina and the organoid. But interestingly, there's more photoreceptors found in the organoids, and maybe slightly fewer of these other neuronal cell types. But another thing to note is that over time, some of the inner cell layers of the retinal organoids die out, potentially because they're not receiving enough oxygen. We're not really sure why, but at this kind of time point, around about day 100, you have all the different retinal cell types that you find in the fetus in these organoids, and they seem to actually mimic both the structure and the gene expression. So the other thing that we're interested in is like, how do the progenitor cells actually differentiate and get to the the differentiated cells? And we can compare how this happens in the fetus, again in the light blue, and the organoid. So again, single cell RNA-seq analysis has shown that as the retinal progenitors or retinal stem cells differentiate into these the final differentiated cell type, they go through a transition state, which can be detected through single cell RNA-seq, and this is this T1 here. So this heat map is showing just a subset of genes expressed, and you can see these are the different cell states. So we have the progenitor state here, the transition state, and the differentiated retinal ganglion cells. And you can see that the progenitor cells are high in SOX2 and vimentin, and this is the case for both progenitor cells from the fetus and from the organoid. And you can see that during differentiation, they go through these changes from kind of high SOX2 through this transition state, which is high for ATO7 and GAD45, and then kind of switch on genes that are associated with the processes of retinal ganglion cells, for example, GAP43 and this RBPMS. So interestingly, both the fetal and the organoid cells also go through these same transitions, right? So you can see that this T1 state is present not only in the fetal cells, but also in the cells from the organoid. So this is the same for other differentiation pathways, and you can see that these developmental gene trajectories are similar in the fetus, the human fetus, and the organoid. Again, saying that these retinal organoids are a really good model, not only for studying kind of the structure of the retina, but also the differentiation pathways and the genes that are important for human retinal differentiation. Okay, so just to sum up this bit, the retinal organoids are good because they're human cell origin, so we can match the exact genetic changes seen in patients with congenital eye disorders. They have the same cell types found in the human retina. 3D arrangement of the cells is very similar. You have these layers, and the timing of development of the organoids and the fetal retina is similar, and also the gene expression changes that happen over time are very similar between the fetal retina and the organoids. What we'd like to do then is try and use these organoids to understand what happens in normal eye development, but also eye development in patients with these eye disorders. So just to go back, we're interested in these eye disorders, microphthalmia, so it's where patients have a small eye, and coloboma, where the morphogenesis, the kind of tissue shape changes, have not finished in these patients. And one of the genes that has been found to cause these disorders is YAP. So YAP is a key regulator of tissue size in many different tissues, not only in development, but also in kind of tissue homeostasis. And it's particularly interesting for our hypothesis because it also responds to mechanical stimuli. So we think that it's a potential gene that might be involved in the feedback between kind of eye morphogenesis and tissue shape changes and cell proliferation, retinal stem cell proliferation and differentiation. So there are these three different reports that kind of describe patients that have YAP mutations and have either one of these or both of these eye disorders. There are also reports in zebrafish where you have a mutation in YAP in zebrafish, which leads to coloboma. Again, you can see this eye in the mutant has not the globe has not closed properly. And in mouse, if you knock out YAP in the eye of mouse, then you lead to this smaller eye phenotype and disruption of the progenitor retinal stem cell cycle and polarity. I just realized, I don't know whether you see my pointer. Do you see my pointer? I'm not sure about this, so I will.
Dr. Manning, yes, we can see that.
We can see. Okay, that's good. That's good. I'll put it on this laser thing anyway. Okay, so we have evidence from not only patients with eye disorders, but also from animal models that YAP is important for eye development and also for morphogenesis and proliferation in the eye. But what we don't really have any understanding of is the mechanisms that YAP regulates to kind of to have these effects, and potentially the effects in the mechanisms in humans themselves. So from in vitro studies of YAP and animal models, we know that YAP is regulated by multiple different inputs. It's part of the hippo pathway, and it's kind of sensitive to how dense cells are cultured. So it's responding to cell-cell contact. It's responding to extracellular signals and also kind of mechanotransduction. So whether cells are cultured on stiff or soft surfaces, this changes the regulation of YAP. And it's downstream of the hippo pathway, which was, as I mentioned, this is a key pathway in kind of regulating tissue size control from Drosophila all the way up to higher vertebrate models that we use in the lab, that are used in the lab. So when the kinases MST1 and 2 and LATs are active, this leads to phosphorylation of YAP and basically keeps YAP out of the nucleus. I should say YAP is a transcription factor, so it's active when it's in the nucleus and it binds to its cofactors TEAD. But on activation of the Hippo pathway, YAP is phosphorylated and kept out of the nucleus. However, when Hippo pathway is off, so these LATs kinases are inactive, they don't phosphorylate YAP, and then YAP is free to enter the nucleus, bind to its co-activators TEAD, and activate target genes. And these are genes involved in the cell cycle and also kind of anti-apoptotic genes. So, as I was saying, YAP requires its cofactor TEAD to bind to DNA and initiate transcription of target genes, but it also has roles in the cytoplasm which are not so fully understood. It's present at the plasma membrane and kind of binds to other proteins at the tight junction, and maybe we don't know whether this is just the kind of role that keeps YAP out of the nucleus or whether it actually has some kind of role here at tight junctions as well. So this remains to be understood. So we have these patients that have a kind of mutations and changes in YAP, but we can also, that are published through PubMed, but we can also look at other online databases to look for other patients that might have variants or changes, mutations in YAP. And so this is to highlight the database. Basically, it's called ClinVar. You can put your gene of interest, whatever gene you're interested in, and then it shows you patients that have had their whole genome or whole exome sequenced, and it highlights the mutations and changes that occur in those genes. So if you want to kind of understand more about the clinical implications of your gene, it's quite a nice website. Okay, so we found 5 different missense mutations. So these are changes where in patients where the amino acid has been changed. So this, for example, this one here, we have got a V to L change at amino acid 55. We also found 2 nonsense mutations. So these are changes where a new stop codon has been introduced. And these were actually from the literature, and we know these are pathogenic and likely causing these disorders. So the thing with whole genome sequencing, whole exome sequencing of patients is that you get a whole load of different changes identified, and you don't really know which ones are actually potentially causing the phenotype of the patient, right? So to try and address this, there have been multiple in silico prediction tools generated. And so we use these different in silico predictions to try and understand which one of these mutations might be deleterious or problematic and might be changing the activity of YAP. So I'm not going to go through all this, but there are various different prediction methods, and we have, for example, here, this prediction method predicts that out of these 6, at least 4 of them are likely to be pathogenic or cause some problem to the YAP function. But really, these are only in silico predictions, so we need to try and test them in cells and see if these are actually do lead to changes in YAP activity. So just to look at their positions on the YAP protein, we have, I've marked these are the missense mutations, so where there's an amino acid change, and these 2 are the ones which introduce the stop codon. And these ones that introduce the stop codon are in this transcriptional activation domain. presumably this is lost or at least truncated in these patients. And we know a fair amount of the YAP structure. We know, for example, this region is the TEAD binding domain, so it's crucial for binding to the transcriptional coactivator leading to transcription. And these WW domains are also involved in protein binding, potentially either in the cytoplasm and in the nucleus. So we think that these mutations found in the patients potentially negatively affect binding to TEAD and so are going to affect the transcriptional activity of YAP. Whereas we're not exactly sure what these mutations might do. We don't know which binding partners they might affect, but they again may have an effect on the transcriptional activity. And so we then want to try and understand the functional consequences of these YAP mutations, as I said, rather than just having kind of in silico predictions, try and do these experiments in cells. So we introduced these mutations into plasmid with YAP fused to GFP, and we then used a luciferase assay. So this is basically looking at how much transcription is happening downstream of YAP in our cells after we've given either wild type or the mutant YAP. So I'll just take you through this graph here. So we start without adding any of YAP. We then add our wild type YAP GFP, and we can increase transcription. If we then add a known mutation that we know will increase transcription, it does that. So this is our positive control. And we also have a negative control where we have a mutation that is known to reduce transcriptional activity, and we see that. So we have our assays working. We can both increase and decrease transcriptional activity. But now we want to know about our the variants, the mutations that we found that have been found in the clinic. How do they affect YAP activity? And even though most of them were predicted by in silico tools to have some kind of deleterious effect on YAP activity, we find that actually only 2 of them are negatively affecting YAP transcription. So as a kind of control, this is not because they're reducing the stability of YAP. So if you do a Western blot against YAP for the cells transfected with our mutant YAPs, we see similar levels between the different mutants. So this decrease in transcriptional activity is not due to a decrease in protein stability, but is in fact like actually some real kind of problem that the mutation is introducing. So we kind of predict that this one, it's in the TEAD binding domain, so we think that this might be affecting binding to TEAD, whereas we're not exactly sure about what this one is doing. So now what we're going to do is look at where the mutant YAPs are localized, whether they're nuclear or cytoplasmic, how they're binding to TEAD, and try and investigate the exact functions of these a little bit more in our cells. But alongside this, we want to try and, as well as looking at these mutations, we want to just kind of have a look at the general role of YAP rather than these specific mutants in human retinal development. And this is where we come back to the retinal organoids. We're using them in the lab, and we can generate, like these pictures were what I showed you before. We're able to generate the retinal organoids with the various different cell populations that we expect. So we looked at whether YAP is expressed in these retinal organoids, and we can see at all kind of stages we get we can detect YAP mRNA, and if we look at protein at the early stages of retinal development, which is what we think is affected in our patients, then we can see YAP is in green. If we look at the kind of zoomed in region here, we can see that actually YAP is found on the in the cytoplasm and potentially at the plasma membrane of these retinal stem cells, which is kind of intriguing because many of the variants that we saw had changes in the domains that are actually going to affect transcription. So we think YAP is probably quite dynamic and shuttling. It's been shown in other cells and in cell culture that YAP is shuttling between the nucleus and the cytoplasm very quickly on the timescale of seconds. So we think that potentially YAP is going into the nucleus here. We can detect expression of target genes, but kind of overall the export of YAP from the nucleus is kind of greater than the import, and so you end up with most expression or most localization in the cytoplasm. So to then try and understand the kind of function of YAP, we have three different approaches. One is to treat these retinal organoids with YAP inhibitor. Another is to introduce the YAP patient mutations into our human stem cells. And a final one is to try and reduce the expression of YAP using CRISPR inhibition. So this will reduce the protein levels of YAP. So these are three different kind of types of manipulations of YAP, and hopefully we'll find kind of similar effects from these three different perturbations. So what I'm going to do now is then kind of talk you through some of the results that we're getting from this, because it's still quite kind of early days and we're actually finding quite a lot of problems. So I'll talk to you about this work. So I'll start with the YAP inhibitor. So if we grow our retinal organoids to day 30, which is when we kind of have a lot of retinal progenitor cells, we then treat our cells with the YAP inhibitor called CA3 at two different concentrations here, and stain for proliferating cells. Then we see them on the outside of the organoid. But in the YAP inhibitor conditions, this is significantly reduced. And so we think that YAP is positively promoting retinal progenitor proliferation. And so that might explain why some of the patients with microphthalmia have YAP mutations, because if there's a decrease in the activity of YAP, then there's maybe less proliferation. And so there's less proliferation of the retinal stem cells, and so you're kind of getting a smaller eye generated. But we'd like to try and understand this mechanism a bit more, so we tried to do some gene expression analysis of these retinal organoids treated with the YAP inhibitor. And I'm not going to take you through all this data, but basically we had real problems replicating data in our retinal organoids. So if you look at this gene, CCND1, so it's cyclin D1, it's a cell cycle gene. We have DMSO treated organoids in blue, and then these two columns are two other concentrations of our YAP inhibitor. And this is looking at gene expression by qPCR, and we can see these are basically three different experiments. And if we look at these different experiments, in the first experiment, cyclin D1 seems to go up in our YAP inhibitor treatment, and in our second experiment, cyclin D1 kind of goes down. And we've seen this like over a couple of, like actually quite a lot of genes, and we're just getting basically fed up with this because it was getting really annoying and we didn't know what was going on. So we started to think, or maybe it's because the organoids are quite heterogeneous, meaning that like each organoid generated has kind of potentially different gene expression. So the organoid in one well of a 96 well plate has different gene expression pattern to the organoid in a different well. So we then looked at this a bit. We pulled together 8 organoids for each sample, because this should kind of reduce the variability a bit. So kind of averaging effects, we tried to reduce that variability. And then we took 8 organoids from one column of a 96 well plate is sample 1. And then sample 2 is 8 organoids taken from literally the next column of the same 96 well plate. So these organoids were treated in the same way. They had the same media changes. They were basically in the same plate in the incubator. And if we look at these 3 different genes, so PAC6 is a retinal progenitor marker. You can see that actually it's got more than 2-fold difference in these 2 samples, even though they were cultured in the same way in the same place. And also another progenitor marker, LHX2, and our kind of gene of interest, YAP. So we've kind of come to the conclusion that the retinal organoids are highly variable from organoid to organoid, and bulk assays where you're essentially mushing up multiple organoids and cells from multiple organoids together are difficult and challenging and give you weird data which are difficult to reproduce. And I think this is why we see a lot of single cell RNA-seq analysis in papers with organoids, because if you're looking at everything on the single cell level, you then cluster cells by similarity, and you kind of avoid any of this like bulk averaging effects that will essentially that are essentially introducing variability. So this is kind of one problem that we're having, in that organoids are particularly variable. There are some labs that are trying to address this by looking at images of organoids through like bright field imaging, and then trying to classify similar stages of organoids, so that we can, without kind of destroying the sample just by taking an image down a bright field kind of a light microscope, we were able to maybe classify these different types of organoids, and maybe that would reduce the variability. But we haven't kind of got to this stage yet in the lab, and so we're at the moment just trying to avoid these bulk assays and use more things like staining and potentially single cell RNA-seq analysis, although these are also quite expensive experiments. Okay, so from the staining, we think that YAP regulates progenitor proliferation, which makes sense with what we see in the clinic and also what we see from mouse models. But we still kind of have some issues here. But then the next thing I want to talk about is trying to perturb YAP protein levels, because this is only affecting the activity of YAP in kind of transcriptional activity, and so only affects kind of one aspect. But here, if we try and knock down YAP using CRISPR inhibition, this will kind of reduce the protein, and so we'll have an effect on YAP activity both in the cytoplasm and in the nucleus, so it will just overall reduce levels of YAP. So to introduce CRISPR interference for those who maybe don't know. So CRISPR interference is not using nuclease activity of Cas9. Instead, it uses dCas9, which stands for dead Cas9. So this basically, it's targeted to any genomic locus using a guide RNA in the same way as kind of classical CRISPR. But when it reaches this DNA locus, instead of introducing mutations or kind of cutting the DNA, this activity is lost because it's dead Cas9. Instead, it's fused to a transcriptional regulator called CRAB. So this is a transcriptional silencer. So wherever dead Cas9 is taken to in the genome, it then kind of silences the transcription in this region. We want to take it to the YAP gene, the transcriptional start site of YAP, where then the CRAB activity would then decrease transcription of YAP, and so would then decrease overall protein levels. And to kind of add to this, we want to make this system inducible so that we can reduce YAP levels at any time that we want during retinal organoid differentiation and kind of production. So to make dead-Cas9 inducible, make the system inducible, we would make it sensitive to doxycycline. So when we add doxycycline, we then get this dead-Cas9 crab expression. But this system is often leaky. So to try and avoid this leakiness, we have a degradation domain so that without TMP, which is another small molecule, this basically leads to the degradation of the dead-Cas9 crab. But when the two small molecules, doxycycline and TMP, are present together, then this stabilizes dead-Cas9, and we would then get gene expression downregulation at our gene of interest, which is YAP. So the plan would be to generate human iPSCs that kind of have this system in, add the guide RNA with lentivirus, grow our organoids, and then induce with doxycycline and TMP our dead-Cas9 crab to activate the kind of cause a YAP knockdown, and we can then do various different methods, for example, single-cell RNA-seq or immunofluorescence, to look at the effect of YAP in our organoids. So we've targeted the construct containing the dead-Cas9 crab to a AAVS1 locus in the human genome, which is a safe harbor locus. So this is known that it's expressed in like multiple different cell types. We've used CRISPR to introduce these to our iPSCs together with a puromycin resistance marker, so that we can kind of select for the cells that actually took up this construct and put it into the genome. This is just kind of a PCR showing that we've got dead-Cas9 in our modified iPSC line, but not in our unmodified iPSCs. And if we use primers that are targeted to the AAVS1 locus, we can find that our modified iPSCs are actually, they have this dead-Cas9 at the right place in the genome. So we're happy that we have this iPSC cell line that can has the CRISPR inhibition system. We can then use doxycycline and TMP to induce our Cas9 expression, whereas when we don't add doxycycline and TMP, there's no Cas9 expression. So this, we can really kind of induce our Cas9. We can then look at a positive control guide RNA for our positive control gene, which is called RAB1A. When we transfect our positive control guide RNA, then we can actually achieve knockdown of our protein RAB1A, and this is our positive control, but we're obviously interested in YAP. So we're testing various different guides. At the moment we've tested 6 different guide RNAs to see how efficient they are at knocking down YAP. They're not quite as efficient as our positive control, but at the moment our guide 6 is the best, and we can achieve a lockdown to about 35% of the original YAP levels. So now we want to see if we can actually knock down YAP in differentiated cells, because actually this was iPSCs. So instead of going for our kind of retinal organoid protocol at the beginning, which is like 30 days minimum, we used a much faster neural differentiation protocol that only takes a week to generate neurons. So we took our CRISPR-inducible iPSC line, we got to day 4 of our differentiation and induced dead-Cas9 with doxycycline and TMP, and stained for dead-Cas9, which you can see in red. So we can induce dead-Cas9 in differentiation, but what you'll see is that we want all of our cells to have our dead-Cas9 to enable knockdown of YAP. However, it's only a relatively small percentage of the cells at day 4. And then when we look later in differentiation, this is even worse. There's a whole load of cells in this field of view, but only 2 of them are expressing our dead Cas9. So we know that we started with a cell population where we could induce dead Cas9 in all of our cells, and yet kind of 7 days after differentiation, it's only a tiny percentage that are still able to express dead Cas9. And this is because, probably because of silencing at our safe harbor locus. And we kind of had a little bit of a inkling that this might happen. So we even included insulator sequences, which are meant to kind of prevent DNA silencing, but we're still getting silencing at this locus. And so it's a bit of a headache, because there's no point in knocking down YAP in a tiny portion of our cells of interest. So we're now trying to look for clones of our cell line, of our iPSCs, but then through differentiation, and hopefully we'll find cells which are, or we'll find clones that are not subject to silencing. But this seems to be a bit of a problem across stem cell differentiation, in that we often, or other labs, speaking to other labs, we find that they're having similar problems, not just with kind of dead Cas9, but other expression systems. So just to kind of highlight, we have these 3 different approaches for trying to find the function of YAP in human retinal development. The 2 that we've tried at the moment are not going super well, but we are currently trying to introduce the YAP patient mutations that seem to have an effect on our luciferase assays, that seem to have an effect on YAP transcriptional activities into iPSCs, and so then put those iPSCs into human retinal organoids. So just to summarize, really, things that I wish I'd known earlier. There's a lot of silencing of transgenes during iPSC differentiation. It seems to be kind of across many different differentiation protocols, and even at genomic even if we put our transgene at a genomic loci that's supposed to be a safe harbor, which is known to be expressed in multiple cell types, and even if we add insulator cell sequencing, we still have issues. And we find that also our retinal organoids are very heterogeneous, which means that kind of single cell analysis is crucial. So this is all to say, like, science is not easy. Think that, oh, we just try some iPSCs and try some new differentiation protocols, but there's always bumps along the way. But overall, what we're trying to do is to investigate the role of YAP in our retinal organoids to come up with a better model for human diseases and human congenital disorders. We've generated a CRISPR iPSC line, which is still good in iPSCs, for example. So you could use this for investigating the function of genes involved in pluripotency. But we have problems then when we kind of start to differentiate. And moving forward, what we'd like to do is to use the zebrafish, which are kind of really good for screening multiple genes. And we try and identify new genes that are associated with eye disorders in the zebrafish, and then go into our human retinal organoid system to try and then see if they're also important genes in human retinal development. And we want to use our models basically to improve patient diagnosis, so testing mutations and variants that have been found in the clinic, but where the clinicians are unsure whether these are actually causing the disorder, and so are testing of these variants would then help improve and kind of define whether these mutations are actually causative in human eye development. And I will leave it there. I'd just like to thank you very much again for the invitation to talk at the workshop. And I hope I haven't kind of presented too much of a negative view of stem cell work, but it's things that I kind of wish I'd known about stem cells. They're amazing, but they're not that easy to deal with, and there are some kind of issues. So I thought that I'd give the issues that we're facing in the lab with them at the moment. And thanks very much for your time.
Thank you very much, Dr. Manning. Yeah, if any participant has got any question, one or two. Okay, so Prashant from.
I'm really sorry, I can't hear. No.
One moment.
You might have to repeat the question.
Yeah, one moment. Yeah.
Hello, ma'am. Now am I?
Yes, I can hear you now.
So whether this YAP mutation can cause unilateral microphthalmia?
So the mutations that we've looked at are either cause coloboma or coloboma and microphthalmia. So is your question specifically about unilateral or kind of generally microphthalmia?
Yes, ma'am. If it is in unilateral, means in which cell this mutation will occur, like retinal progenitor. If it is in retinal progenitor, means it should be bilateral, ma'am. How it is restricted?
Yeah, no, this is a really interesting thing, because a lot of the time also in eye disorders that there's a parent, for example, that carries the mutation and is not affected, but the child carries the mutation and is affected. And so you have this thing called incomplete penetrance. And so this can be across two eyes, unilateral versus bilateral, because the mutation is present in the germline, right? It's present in every cell, but yet somehow we really don't understand why only one eye is affected, and also potentially only one of the family is affected, even though they both share the same mutation. So I think the current thinking is with the kind of parent-child thing is that maybe there are other multiple genes required to kind of result in these microphthalmia or coloboma. But the unilateral bilateral thing is a real mystery, and I think it kind of shows the randomness and the stochasticity of development. However, although kind of eye development must be stochastic somehow, it's still very reproducible, and so we really don't have a good answer as to why sometimes you only have like a defect or a small eye in one side and not both. It's a great question, but we don't know the answer.
Is there any role of defect in bone morphogenesis may cause this defects in closer or?
Sorry, what was that? Sorry.
Is there any chance for defect in bone morphogenesis may cause these conditions?
Bone morphogenesis. I think these disorders are mainly of the eye cells that we see at the moment, because the bone are still quite malleable at this time and not really calcified. So I think it's probably still within the eye tissue where the problem is occurring.
Okay.
Thank you.
Prashant.
Thank you.
Is there anybody else? Okay. So I hope that one day the organoids would be able to see the things. I mean,
Yep.
as a functional eye that we would expect otherwise.
Yeah.
Okay.
Yeah, no, it's true. I didn't actually present that the organoids are light sensitive, but it would be amazing to have a fully functional organoid that can be maybe transplanted or
Dr. Manning, this is an fantastic opportunity to interact with you. And on behalf of the all participants, not only that, we have got to know a bit of your work because of Srishti's presence in your laboratory.
Yeah.
We wanted to thank you for everything. And maybe it's a beginning of a new interaction, a new collaboration maybe in the future for that. Okay.
Yeah.
Yeah.
Absolutely. Yeah.
Yeah, we would like to thank you for making this time and making yourself available for the participants in this stem cell workshop. Thank you so much again. Let's give a.
Thank you very much.
So we'll close here because we are running out of the time.
Yeah.
We'll close here today. Thank you very much again.